So we'll move on now to the round table. Segev, maybe you take the one. I think that you are muted, Segev.
Yes. So sorry. We heard many great talks now about the use of mouse models to study autism, and we'll now in the roundtable we'll have a discussion about the differences between and similarities between humans and mice. We have with us four distinguished scientists. Professor Idan Segev from the Hebrew University of Jerusalem, Professor Illana Gozes from Tel Aviv University. We have Professor Ornoi from the Hebrew University Hadassah Medical School, and also Dr. Idan from Bar-Ilan University. And each of our guests will talk on a different question that we gave them beforehand. But unfortunately, we're not going to have time for questions from the audience. And so we're going to start with Professor Idan Segev, and he will talk about the following question. Can we use the mouse brain as a model for human brain structure, circuitry, and physiology? And if we can use it, what shall we be worried about when we do such experiments? Thank you.
Thank you. Can you hear me?
Yes.
Okay, thank you very much and welcome. Good. Thank you for inviting me. So I have a few points to make. It's a discussion. I'm not an expert in mice, but one thing I should say, and I think already John Steinberg, the great novelist who wrote a play called Of Mice and Men in 1937, he already realized then, as many artists already do before scientists, that there are, of course, similarities between the mice and the men. But the question is whether these similarities are sufficient, really, to become so important that we cannot even do a research on human without comparing ourselves to mice. It became so embedded in our brain that, because we know so much about mice naturally, because you can manipulate it genetically and all these things, and you have, ethical issues that are easier, that we cannot even anymore study the human without comparing ourselves to mice. This must mean that we believe that we are close to mice, that we are similar to mice, which is, I guess it's true in some sense, but we're similar to any other animal. So that's one issue that I wanted to raise, the issue of whether we are similar to mice. To what extent are we similar to mice, and what can we learn from being similar to mice? So maybe we should get rid of this similarity and start to directly, not comparing, but directly studying human. So that's my first comment. The other comment about human is, and that's why Segev invited me, because we are studying human in my lab and other labs in the world. We are studying human at the cellular level, synaptic level, genetic level, in live tissue, following brain operation. So we now can take things that were usually thrown to the garbage, post-operation tissue of human, let's say cortex or hippocampus, and use it as a tissue, as we did for mice so many years, to study the circuitry, to study the synapses, to study the cell properties. And this is the technology that is coming more and more into the science. So about 20 labs in the world now are studying directly post-operation brain tissues. You can discuss whether it's healthy or not healthy and all these things, but this is for another time. And while discussing it and while looking at it myself as a modeler, you certainly can see that we cannot really say that our brain is just an extension at a single cell level. It's not an extension, just linear expansion of what happened to the mice. For example, layer 2-3 in the cortex of human is much, much more thicker relatively to other layers, and in the mouse it's not the case. So something must be very important in this circuitry, layer 2-3. Single cells, you cannot just take a mouse cell and extend it and it will become a human cell. Human cells are much more complicated in terms of bifurcation, number of synapses, properties of individual synapses. So the question is really, again, Of course, I need to continue studying mice. That's what I can do until today. But I think it's time to a little bit go out of the mouse, give up this similarity and study directly human, as you do genetically, but we can do it physiologically today. We can discuss it later if we have time, but that's my little speech about the preference of studying directly human without comparing it to another animal.
Wonderful. Thank you very, very much. And let's move to Professor Ornoi, who can tell us something about the next question, which is, can we use mouse social behavior as a model for human social behavior? And if so, for which aspects? Of course, we all think about language when we think about that. Please.
This is, I would think, a very interesting example of what I call double translation. We have behavioral phenomena in human. We want to see if we can see similar things in mice. So this is translation from human to the mouse. And then we develop tests to test exactly these behaviors. And then we ask the question, okay, we have done that in the mouse. How similar is that to the human situation? And I think that studying behavior is very strange in that respect. Now, when we are talking about social behavior, it is composed of many different issues. And when we look at the mouse, we would prefer to test these specific different issues separately. And this is what is really done. We can test anxiety, anhedonia, despair, apathy, etc., because there are very good tests developed, and these tests actually are repetitive. You can test the same animal, the same mouse, again and again. Of course, you have to obey some laws or rules of testing and see that the results are similar. Now, is that exactly what occurs in children or in adult human? We believe yes. Is that correct? We don't know. However, in many cases, if we take, for example, depression, and we have mouse models of depression, we can make that assumption much stronger when we use drugs that are used in human to eliminate some of the symptoms, and we can see that the same symptoms in the mouse are also eliminated. When we can do that, and we do it repeatedly in mouse models, not of autism, because we don't have the cure or the treatment, so-called, but if we do it for models of depression, we can really see and actually require that a good model would also be a mouse where we can eliminate the symptoms of depression. So if I want to summarize, we are taking human situation, human behavior, translate it into the mouse, look for tests in the mouse, and they have to be quite accurate, and ask additional questions. How would the mouse behave when we treat him? And then go back to human. And if this is fulfilled, I personally think that we can do it. But is that a correct answer? I would open it to the audience. Thank you.
Thank you very, very much. We're going to move now to Prof.Illana Gozes, and she will talk about another question which is related to this session, which is, shall we expect similar sex-dependent differences between humans and animal models, given what we heard already that autism is male biased?
So my answer is yes. But we have to keep in mind, like Idan already told us, and we are all in agreement, that mice are not men. And so, and you know, the estrous cycle in a female mouse is 4 days, and it is month in us, in women. So these are differences. But on the other hand, both the mice and the humans, they have X and Y chromosomes. And I want to say, and this is kind of important, we have genes that are very conserved between mouse and men. For example, a gene that is very close to my heart, and you heard my students speaking, is ADNP, and it is 90% similar, identical between mouse and men. And actually, when we looked at the mice originally, we actually detected autism much before it was discovered that it is mutated. So this is something to keep in mind. And for a long time, as researchers doing behavior in animals, we only worked with males because the females have an estrous cycle, so they wouldn't behave the same. So we kind of ignored them. But what is now demanded is actually to study both. In terms of how will it behave, again, because of the estrous cycle, then there probably will be differences. But as we heard, also in the plenary lecture, we heard about the glia having a great effect. And I think all this can be dissected much more simply in a mouse model and then translated to the human situation. And you can't study behavior in a tissue. You have to have a model, and also you can't translate. I mean, if you want to develop a drug, you can't translate to human. You can't test the drugs directly in children. You have to go through a model. And the mouse, it's limited. We know it's limited, and the behavior is going to be limited. But and of course, the social behavior and all the behaviors are very limited. But we heard that these are things that are being developed, and I think we must use the models to go along. And we can also use other models, like for speech, we can use the birds, and there the canaries, they sing, the males are singing, not the females, but we can use other models as well. But we have to be careful to see that our genes are really telling us something. Because like for an Alzheimer's models, for example, the amyloid beta precursor protein is not the same in mouse and men, and this is a very great difficulties in my mind. So this is not, it's also sexually dependent, actually, because there is more Alzheimer's in females. So thank you very much.
Thank you very much for this message. And we're going to move now to Dr. Evan Elliott, and he will speak about another question which is related to the interaction between the environment and the genetics. And the question is, can we use mice to model the interaction with the environment, such as the exposure of drugs or effect of the microbiome in the context of autism?
Thank you very much. I think when thinking about this question, it's very good or useful to compare it to studying genetic predisposition. And some of the challenges of looking at environmental predisposition to autism. So one of the challenges is the fact that environmental factors, as well as we know them, tend to have a much lower penetrance for increasing the case of autism. So for example, if there's a particular environmental factor which may increase incidence, it might do it by a very incremental amount. This is different than, let's say, someone who has a mutation in SHANK3, which is much more likely to induce the autism phenotype. And therefore, you have to keep in mind that therefore in modeling, you may need, first of all, much higher numbers of animals to get really robust effects, because you expect both in terms of molecular biology and in terms of behavior for the effects to be more incremental or smaller. But even more important than that is the idea of modeling environmental interactions. So what the interesting question would be to see the differences between those that actually do show or that do react to the environmental factor and to those that do not. So to give an example, when often giving one example of environmental factor that you often give is giving the maternal immune activation, which is mentioned in one of the previous lectures, where it can cause maternal immune activation in a pregnant mother. This increases their immune response during pregnancy and may increase the chance of autism-like phenotypes in the offspring. So when doing that, very many labs have shown, and we've seen this as well, that you get sort of a bimodal distribution. In other words, in the offspring, there are some that do not show any behavioral phenotype, some that show a very best phenotype. There's not much in between. And so the question is, okay, so what's causing this predisposition? Why do these mice react and these do not? And I think that's one of the more interesting questions when looking at environmental modeling of modeling of environmental factors in autism. Because very much, even 5-10 years ago, what usually happened is that they would look at those mice that show a robust phenotype and study them, and those that didn't, well, they didn't study them as much. But actually, that's even more important to look at the differences in terms of resiliency versus susceptibility. Another important factor to keep in mind is, can we even model it, is to keep in mind that you have to translate the environmental factor from the human world to the mouse world. So for example, there's some environmental factors which might happen in the first trimester of pregnancy in humans. Well, the first trimester of pregnancy in mice might be different in terms of time points within pregnancy. And so you very much have to keep that in mind, and people sometimes forget that. So for example, the third trimester is pretty much like the first week after birth in a mouse. And so in translating biologically, you have to keep that in mind. I would also mention, just as we're talking about sex-specific effects, that I found that the environmental factors in modeling very has strong sex-specific effects. You'll often see effects in mice and males you do not see in females. My tendency to think, I don't have enough empiric evidence to back this up too much, but my hypothesis is that the environmental factors might even be more sex-specific than the genetic factors, but I'd have to back that up somehow. One last thing when talking about the microbiome. So the microbiome is it's something which varies very much from species. So if I want to model a microbiome in humans to mouse, and let's try a probiotic, so you have to see, first of all, does that probiotic, does that microbiome even colonize a mouse? So just give an example. They've been using 2 probiotics in mice species. We found that one of them colonizes very well in mice, and one of them does not. And the mice, it's just like the probiotic just goes through the system and goes out, which is not the case in humans. So there's very much a species-specific effect that has to be controlled for and kept in mind when looking at the microbiome. Those are my major points that I thought to bring up.
Shlomo, do you want to summarize?
Yeah, I was muted, sorry. And so I would like to thank the speakers for inspiring talks and discussion, and we just touched upon the important questions that we have raised, and I hope that this discussion will keep on here and there in the future, and I hope that we'll meet each other physically in the next session. So thank you very much.
Yeah, thank you very much for all the speakers and for the roundtable.
Bye-bye.